So this is our BRIC-20 SVT, which is the science verification test. We were down at Kennedy to integrate our science. This is our building, and Darren carrying the stuff into the labs. This is the space shuttle processing facility. This is the hallway, and we thought it was funny, the most colorful thing in the hallway were the signs that told you caution where the doors would swing. This is inside our lab. So this will be our lab, the laminar flow hood for the sterile processing, and those classic refrigerators, just basically a standard wet lab. And what you see here are our specimens. These are bottles of media, and here I am just looking at them, and the seeds are in the little petri dishes there. So this is a sign on the door, pretty straightforward. Now we're in the central services where we are microwaving our media to melt it down so that we can pour the plates. And a pic of me just standing there taking pictures in the facility. So here's Darren again getting our media out. And now we're going to pour the plates. It's a secret little place. Okay, so now we're at the laminar flow hood. We have the plates out, and Darren's starting to pour the plates. And of course, he even let me pour a few, just to prove I could. And we're done. There they are. Now they've got to dry a little bit and solidify, so we're going to take a break and go to lunch. After lunch, here we are again. We're ready to plate the seeds. The seeds are on these little white filter papers, as you can see there. We've got them all sterilized. We brought them down to Kennedy sterilized and on the filter papers. So what you see me doing is I'm basically just taking the filter paper and squiggling it back and forth in my hand to deliver the seeds onto the plates. That's a scientific term, by the way, squiggling it between my hands. So there you've got a close-up of the seeds on the plates. And there the seeds are. They're all plated out, ready to go. And then we wrap them all up because they're going to go into the cold. And there's our final seeds on the plates already, and they're going to go into the refrigerator overnight. So we're leaving again. Next morning we came in, we laid them all out and gave them an hour's light treatment. That was to try to be sure that all of the germination would be synchronized. Nice little close-up of them. And down to the hardware lab. So now we're going to get to integrate them into the PDFUs. And when I say we, I actually mean the technicians at NASA. That was Darren that you just saw, all geared up in sterile conditions. And this is Donnie, our technician, putting things together. So this is the PDFU, and he's putting it together. He's assembling all the parts. Now he's going to actually integrate the science. The little petri dish at his right hand, and now it just went into the PDFU. There's number 1, done. Number 2, and number 3. Number 4. And finally, number 5. So that's one of our bricks complete. Now what happens once those are done is we're going to load the RNA later into the chambers so they're ready. So this is the RNA later being pushed into the chamber in the PDFU. This is our fixative. So once we're on flight, they'll actually inject that fixative into the plate. Right now it's just in a holding chamber inside the PDFU. Now they're tightening down all the bolts. And we do another one. Obviously, we repeat the process until all 5 of them are complete. And those are our bricks. And there you have it, integrating the science.